Good afternoon to all of you. At around 3:15, I think that practically nobody was here. I remember when I was small in Mauritius, whenever we used to go to see movies, when you don't have sufficient number of people, you don't have a critical mass, we used to say séance relâche. So they won't play the movie. I'm sure my friend Dr. Gopi will know about that. So we thought that maybe we'll have a séance relâche, but I'm happy to see all of you here, to have taken your precious time in a time where people are doing shopping for Diwali and so many things going on at home, and you've taken your precious time to come here. So I'll try to do justice by just sharing a few things which I know with you. So today's topic is astrochemistry. So as some of you may know, research is becoming more and more multidisciplinary. Why do people do research? Because there's so much we don't know. The more we know, the more we realize how much we don't know. Now, in each topic, you have certain issues, certain puzzles which have not been solved. Like if you take astronomy, if you want to know about the origin of the universe, origin of stars and galaxies, there are issues which are not clear. That's why people do research. So during the last 20-25 years, I would say, we researchers, we have found that it's very important to go multidisciplinary. What do I mean go multidisciplinary? It is to inspire ourselves from other branches of research and see what's being done in other branches and collaborate with those researchers and take those ideas and concepts on board to be able to solve those issues and puzzles which I've been talking. So astrochemistry is a topic which has existed since nearly 20-25 years. So a group of astronomers and a group of chemists, I think started in some university in US. They realized that if they want to study about molecules in space, we cannot have researchers only in astronomy. So they realized it is important to have a major input from people in chemistry. So it's a collaboration between chemists and astronomers and also physicists, which have led to that new field called astrochemistry. So before going through the slides, first I would like to apologize. This is a talk I gave in an international conference for 1 hour, and during the last 2 days I had a very hectic time in a very important conference on the SK project. I didn't have time to remove the extra slides. So I apologize for some slides. I'll just go through very fast. But don't worry, I will try to explain as much as possible the slides which are relevant to this audience. Before starting, I would like to introduce one of my colleagues, Sabrina. Can you please stand up? So she's my colleague in the Faculty of Science. Can we consider yourself to be a molecular biologist?
Yes.
Okay. So Sabrina has worked, there's another topic called astrobiology. So when she was in US, she was working in US for some time, she did do some research in astrobiology. So I would like to thank Sabrina. I took some of her slides for my talk. Anyway, you already know about that. So this will be the contents of my talk. And some of them I will go a bit deeper, some I will just scan through very fast. So I'll give you an introduction about astrochemistry. And I will elaborate what are prebiotic molecules, how they are related to DNA and RNA, and the molecules which we have detected in space. Whenever I say we, it's a whole community of researchers in astronomy. It's not myself, but it's a whole community. And where do we see those molecules in space? And you'll be interested to know that those molecules have not been detected only in our galaxy, but they have been detected in neighboring galaxies also. And the origin of the spectral lines, so those molecules have a signature. Each molecule has a very specific signature, and we astronomers, we try to look for that signature to be able to conclude that those molecules do exist. And then, Kamlesh, you said, should I divide my talk in 2 parts, or I just go? Okay. So Kamlesh feels that in order not to burden you too much, it's good to have a break. So maybe I'm taking the decision now. I'll take the break after the origin of the spectral lines. And maybe I can take some questions. So I'll call it at the first part. The second part will be, what instruments do we use to detect those molecules? How can we conclude that those molecules do exist in space? And I'll elaborate on some of the telescopes. I will tell you briefly the different kinds of telescopes we have, and what are the main telescopes which have been used to detect those molecules. And I won't go into the details of the detection. It's a bit, I won't say complicated, but it's more of electronics and hardware, which I won't elaborate in this talk. And then I will talk briefly about the SK project. So SK project is going to be the biggest telescope in the world, which is going to be built in Africa, and it's going to be the biggest scientific project in Africa. It's a project which will have a lot of spin-offs in technology, in big data, machine learning, machine language. I can see David smiling at me. David is doing his undergrad project with me on big data, machine learning, and so SK project, that huge amount of data, you need to use big data. So as I already said, astrochemistry is a multidisciplinary branch, which is a combination of astrophysics and chemistry. So basically what we do in astrochemistry, we try to use different types of telescopes to observe what we call spectral lines in space. So we can have radio telescopes, we can have millimeter telescopes, infrared telescopes, and optical telescopes. So we use those telescopes to detect molecules in space. So what are the kind of molecules which are of interest to us? It's molecules which are at the basis of cells, which we know, which comprise our body and all living things, about DNA. And we try to understand how those molecules could have been formed in space. So you have different explanations by different groups of researchers, and each school of thought has its own pros and cons, and we are quite far to understand exactly the procedure, the scientific procedure, how those molecules have been formed. By the way, I'm basically a radio astronomer. Usually, most researchers during their life, they choose different topics of research. So astrochemistry is a new topic to me. It's only, I would say, 5 years. And the more I'm learning in astrochemistry, the more difficult I'm finding it. So I'm definitely not an expert in astrochemistry. I'm just come here to share what I know with you. So those molecules are found in different regions in space. So just briefly to tell you, to set you can have a 3D picture in your mind. Just imagine a galaxy. So what is a galaxy? It's a galaxy can have a few billions to a few hundred billions of stars. Like our galaxy, the Milky Way, has a few hundred billions of stars. And you have different types of stars. So when we look at stars, we look at different types of stars at different stages of evolution. So some molecules are found in the interstellar medium, that is between the stars, the space between the stars, in some regions which are very rich in hydrogen molecules and H plus and H atoms. And we can also see the molecules in comets. So we can see comets very rarely when they come very close to the sun. Because of the interaction between the sun and the comet, we are able to see the comet, and we can point our telescope to the comets and see them. We can also see the molecules around certain stars, which we call circumstellar regions. So during the last 25 years, we have pointed our telescopes in different regions in the sky. And we know, we astronomers, we know in which regions where we can find molecules. So you can have two kinds of observation, which we call, first one is a targeted observation. We know in such a region like molecular clouds, we know you're bound to see molecules. So you can point your telescope there and try to discover new molecules. But you can also, like it's becoming more and more common among astronomers, you can do what we call a blind search. And blind search can lead to serendipitous discoveries. You know the meaning of serendipitous? Maybe in case some of you don't know. Something is like penicillin, discovered by accident. And during the last 10 years, it has been really wonderful to see that in regions where we never thought molecules would exist, in many of those blind searches, we have detected molecules, which is something really fascinating. Kamlesh, I would advise you to check on my time because I can be carried away on each slide. You feel I'm going too slow, you can. So basically astrochemistry is about studying the chemical complexity of the universe. We would like to know all the different types of molecules which we know on Earth, and we try to detect those molecules in space. Now, when we use a telescope, just something briefly I should tell you about telescopes. There are two very important characteristics about telescopes. First, it is the sensitivity, which is our ability to detect weak emission. So most telescopes right now, they can see up to some distance in the universe. They cannot see further because of the limitation of the sensitivity. So the SK will have 100 times or 200 times better sensitivity, which means we will be able to see maybe further away, closer to the Big Bang, and molecules which we have not detected before. And secondly, there's something called resolving power. So you have two types of resolving power. It's ability to distinguish between two objects in the sky. Like if you take my two fingers right now, if I go 1 kilometer back, you'll see only one finger. So the ability to distinguish between two objects in the sky is called resolving power. Now, in astrochemistry, in the detection of molecules, the resolving power is not so important. But what is important, it is the frequency resolution. If I take just 20 molecules, I can do calculations using quantum mechanics to find out the different lines which will be emitted by each molecule. I can find 1,000 lines. And all those lines are very close to each other. So if you want to be able to detect a particular line, your telescope must have what we call a very good frequency resolution. So maybe some molecules have not been detected so far because the frequency resolution of our telescopes are not sufficiently good. But definitely the SK project will enhance this capability. So why is it so interesting to observe molecules in space? Because there is so much about the interstellar medium as an intergalactic medium and formation of stars, how the first stars formed, how the first galaxies formed. There's so much about the galaxy, so many parameters which are not so well known, where the molecules can tell us more about the things which are not so well known. So I will just skip that. This is just what are the main goals of astrophysics? Yeah. So this is a timeline about the history of the universe. So the universe started somewhere here. So universe exists around. Thank you. So the universe exists for nearly 14 billion years. My aim of my talk today is not to talk about what happened here, but just to tell you, initially there were some particles called quarks, and then leptons and hadrons formed, and it's much later in the historical evolution of the universe that atoms form. So the first atom is obviously hydrogen. You just can imagine a proton, which is made up of 3 quarks, and an electron around. Now, you had a small percentage of helium also, and all elements, which we know, all elements in your body right now, most of them, apart hydrogen and some helium, have been produced by the process of nuclear fusion inside stars. And they have been formed mainly because the universal constants have the values they have. One can imagine a different universe where the universal constants were different, and it would have been a different universe. So all the elements, not all, but most of the elements have been formed inside stars, and the molecules came after. So we astrophysicists, we want to know, starting from the elements, and when I'm talking about molecules here, it's mostly carbon-based molecules, because we are more interested in molecules which comprise the DNA and other living things. So what we don't know for sure right now is how the molecules formed, and the molecules did not form in one stage. You have different stages. So we astronomers are not very clear how many stages for any molecule for it to be formed, and what are the different steps, and what are the conditions prevailing in the interstellar medium or around stars which enhance that formation. There are different theories, there are different schools of thought. So why are we not so sure? Do you know why we are not so sure? Because we are limited by observations. Our telescopes are limited. More powerful the telescopes, better sensitivity, better resolving power, and better frequency resolution, then our theories will be more fine-tuned. But nevertheless, different groups of astronomers and chemists, through the umbrella of astrochemistry, have written different theories to try to explain how those molecules have been formed. So basically what I want to tell you, it is still a mystery. Now I come with some slides of my colleague Sabrina, which I won't take long because that's not my specialty. So the aim of my talk today is not to elaborate on prokaryotes and eukaryotes, but just to show you that they were among the first units to be formed in the universe, and whatever more complex molecules, more complex organisms or living things were formed much after. So this is a nice tree which I got from Sabrina. Thanks a lot, Sabrina. Don't worry, I'm not going to elaborate on that. But there's one thing I forgot which you told me, which I learned from you. At some stage there was some kind of mixing between the bacteria and archaea. And if that mixing was, so I remember well. So because if that mixing was not there, we would not have had this branch, and maybe we would not have existed. Okay, that's the only thing I remember. Everything else she told me I've completely forgot, but that was not my field. So basically, so this tree, it's basically falls under astrochemistry and astrobiology. Okay, so astrochemistry is more about the molecules, but astrobiology is more about focusing on the different aspects here. So we want to know what is the last universal common ancestor, which we call LUCA. So this has come much after the molecules. So you had the first, I would say, primary molecules, secondary molecules, which are more complex. Gradually, the molecules evolve in complexity to reach LUCA, and all other organisms which we know on Earth were formed after. So if you want to understand this better, it's very important to understand how the molecules form up to the LUCA, and which we still don't know exactly what it is. So this is just to give you an idea about the different units or organisms which existed on Earth. Like I've told you, the universe is around 14 billion years. And out of 14 billion years, we exist. The humans have evolved only during the last 1.8 million years, which is insignificantly small compared to the age of the universe. So what happened before? It was basically formation of those molecules and evolutions of the molecules in complexity. So you can see here the hominid species is just 3 million years ago. And the prokaryotic groups, it's nearly 4 billion years. And the Earth was formed only 4.5 billion years. So just to give you an idea of the kind of, my aim is not to explain this slide here. This is just to give you an idea about the kind of time scale we're talking about. So the molecules had nearly 4 billion years to evolve to whatever it is right now. So people have tried to create those molecules in the lab through the Miller experiment. So there are other experiments also where scientists have tried to produce those prebiotic molecules in the lab. I won't elaborate on the Miller experiment, but just to tell you that this has been done. So all living organisms are dependent on 3 types of very large molecules. Okay, so they are DNA, RNA, and proteins. So we classify them as biological macromolecules. And as most of you know, without DNA, RNA, and proteins, no known forms of life could exist. But I'll just make a small parenthesis here. It could be, during the last 5-10 years, people have been talking about other forms of life. So why do we need a carbon-based form of life? But this topic will not be discussed today unless you want to discuss it after. So I'll just go fast through the structure of DNA and RNA without any explanation. So basically they have a nitrogenous base, a pentose ring, and a phosphate moiety. So I won't describe the molecules. I'm just putting the slides just to show the kind of molecules which we try to detect in space. Yeah, before I continue, what I can say is many of those molecules are very unstable and cannot be produced in the lab. So the advantage of observing such molecules in space is because in space you have very extreme kind of conditions, like temperature, magnetic field, and you have the time factor also, which can be billions of years to enable such molecules to be formed. So many of those molecules are unstable in the lab, which means you cannot study about the spectra, but we can see them in outer space. And some of them, they cannot, because again of the instability, as soon as they are produced in the lab, they disintegrate. So that's where a branch of chemistry is, which is called computational chemistry. And one of my colleagues at the University of Mauritius, called Professor Ramasamy. So actually it's a collaboration between him and myself, where an astrochemistry group has emerged at the university. So basically what he does, he tries to find the different characteristics of molecules by using computation. So he's got a supercomputer. Madina, can you correct me for that? So she's doing her project in astrochemistry. So you still use that supercomputer in US? Okay. So they use a theory in physics called quantum mechanics to try to find the energy levels and the spectral lines and some other parameters of those molecules. So the beauty of computational chemistry is that you can find so many things about, so many characteristics of molecules which could not have been found in the lab because they are unstable. What kind of work which Professor Ramasamy and myself are doing is we try to scan the list of molecules which have been discovered, and we try to, with arguments of course, to predict other molecules which have not been discovered so far. And we try to look at the different conditions prevailing in space and see where they can be found. Then what we do is we use that supercomputer in US to do some very complicated calculations, which is very time consuming, which an ordinary computer could not do. So we use that supercomputer to get the energy levels associated with those different molecules, which is very difficult to do if you just use an analytical method. Then we know what frequencies to observe, and the next stage, which we have not done yet, is to ask for telescope time to try to observe those lines. So our dream is to be able to detect some molecules which have not been detected so far. But it's good to dream. Dreams are the best part of life. So again, I won't elaborate, just to give you an idea what kind of molecules I'm talking about. So those in molecular biology and medical doctors will be very familiar with those molecules. The DNA nucleotides. So what is fascinating is those molecules are very complex, and for quite a long time we thought that those molecules existed only in living things on Earth. So today we can say since the last 20-25 years we have very complicated molecules existing in space. Now I won't make any conclusion, but it's up to you to make your own conclusions. It's basically, if I'm in a science fiction movie, the rate at which the molecules are being detected and the kind of increasing in complexity of the molecules which are being detected, and the advance of technology, the way it is going, it looks like at some stage, I don't know what's going to be 25 years or 50 years or 100 years, at some stage we would have discovered all molecules in outer space. I might be philosophically wrong, but that's my way of looking at it. So as I said, some slides I won't describe, just to have an idea about what molecules I'm talking about. So this is a list of common amino acids. And as you know, amino acids are very important to understand living things, cells. So most of them have been discovered in space. So here I'm going through the basics of some amino acids. I can understand why you can see all those molecules in space, because if you take the time of billions of years, okay? So if you take the evolution of the universe, evolution of stars, evolution of galaxies for billions of years, you would definitely encounter most kinds of conditions which you can imagine. Conditions, I'm talking about temperature, the density of hydrogen, the kind of magnetic field which could have existed. It looks like from some latest papers, magnetic fields also are very important. So because you had all those kinds of conditions, and you can easily think that it should be possible for those molecules to be formed under different conditions. So in chemistry you have L and D isomer depending on the geometry. So what's interesting is they are the same molecule in terms of structure, but when you look at the spectral lines, they are very different. So by observing certain specific spectral lines, one can say what kind of isomer you are looking at. So many of you know that when you talk about carbon, about the benzene ring and cyclic structures, so many benzene rings, molecules where you have one or more benzene rings and cyclostructures have been detected in space. I will talk maybe about it later, just in case I've forgotten about it. There are some molecules called fullerenes. Their structure is C60. You can have 60 carbon atoms, or you can have C70. They basically look like a ball. It's like a ball when they are interconnected. Now, try to imagine how can such a complicated but at the same time symmetrical molecule have been formed in space. So I just skipped on some more molecules which we had. So here I'm giving you some molecules which have been, some of the first molecules which have been detected in space. Yeah, I did not forget about it. So C70 and C60. Those are among the latest molecules which have been detected. You can imagine a ball, okay, with you can have 6 sides or 5 sides, and those molecules have been detected. Well, what is interesting, some time back, my colleague, Professor Ramasamy, we had a Skype conference with a Nobel Prize in the field. So we're talking, he was talking about Leonardo da Vinci. So why he talked about Leonardo da Vinci? Because, as you know, Leonardo da Vinci was a genius. If we go back to the time of the Greeks, where we had Pythagoras, Socrates, and Plato, it was a holistic way of learning. One cannot be a good physicist if he is not a good musician also. So you have to be a good musician, a good philosopher, a good physicist. That's what I call a holistic approach towards research. But from what has been done recently in some topics in astrophysics, it looks like we have to become holistic again. The holistic part of research is becoming more important. One has to try to inspire yourselves more from different fields. It can be a field which is completely different. Yeah, while I was talking about Leonardo da Vinci, he did not know anything about astrochemistry or fullerenes. So in some of his paintings, or I don't know whether it was paintings or actually, yeah, but I forgot what kind of artistic thing he used. He did draw the C60 and C70. So it looks like the fullerenes were already existing in our mind. One just had to discover it. Now, the process of detecting a molecule, it's not like you point your telescope. Sorry. You point your telescope towards some circumstellar region or some region in the interstellar medium, and the next day you say, I've discovered a molecule. So just to tell you that it's not an easy process. Sometimes it can take 1 year, 2 years, 3 years, 5 years, or 10 years. So the observation has to be repeatable, okay? And there are so many factors which you have to consider. Whether the observation has been polluted by some other hardware factor or something wrong in the software, and some calculation which has been done wrongly. But those molecules have, so basically there's a transition stage which we call some molecules are reported but need confirmation. So those are the molecules which are still waiting for confirmation. So what it means, it has to be observed by other telescopes, and there are certain spurious features in the observation which have not been elucidated yet. So besides molecules, one can also have spectral lines due to some different reasons. Let me take the example of water. So it is possible for water as a gas to be in a given energy level, and we have what we call an inverse population. It changes the energy level suddenly, and it emits a very strong line. So this is what we call astrophysical masers. So those are not molecules, but spectral lines which are very important in astronomy. And those lines, even the lines emitted by the molecules, many of them are found between infrared and radio. So nowadays we have different kinds of telescopes. We have gamma ray telescopes, X-ray telescopes, ultraviolet telescopes, optical telescopes, which all of you know. We also have infrared telescopes and radio telescopes. The SK project is going to be a radio telescope. Now there's a region between infrared and radio, which we call millimeter telescopes. So millimeter telescopes are very special, and if I'm not mistaken, more than 50% of the molecules are detected in the millimeter region. As I always tell my physics students, reality is not restricted to our senses. Okay? There's so much there in the universe which our eyes cannot see, but which our telescopes can see. So this is a picture of the universe made by a radio telescope. It's at 21 centimeter, which corresponds to a frequency of 1420 megahertz. So the reason behind this line is, so you can imagine a hydrogen atom where you have a proton and electron. So the electron has a property called a spin. So when the spin changes, let's say, you can imagine it to be this way, it's not exactly this way. When the spin changes, there's a change of energy, and that change energy produces a photon of 21 centimeter. Now, since most of the mass in the universe is hydrogen, when you're mapping hydrogen in the universe, you're actually mapping the distribution of matter in the universe. So this is basically distribution of matter in the universe. So this is basically the galactic center, and the rest are extragalactic objects. So this is like, this is carbon monoxide. So you can imagine some spectral line on carbon monoxide. So you can tune your telescope to look at that line and look at the distribution of carbon monoxide in our galaxy. So you can see the galactic center here. It's as if our eyes become sensitive to that particular line, which is emitted by carbon monoxide. This is a radio map of the universe at 408 megahertz. Now, if you look at the universe at different wavelengths or different frequency, it is different. Nowadays, if you want to do research in astrophysics, even astrochemistry, it's very important to go multi-wavelengths. What do I mean? Suppose you have a galaxy or a given region which you want to study. If you look at it only in one wavelength, it will be difficult to say, to tell any story, if you want to call it a story, about this object or region. It is important to look at it at as many wavelengths and get as much information about the object. Then the probability of your people to believe your story will be very high. Otherwise, you will tell a story just like telling stories to kids, and people will not believe you. So I did mention a bit about it in the beginning. Those are the regions in the Milky Way, our galaxy, where molecules have been detected. So you can see comets, dense cold interstellar clouds, some regions around certain stars. Basically, the regions which you have to look for to find molecules, I would say, are pretty well known by astronomers. See, I'm just giving different phases of the interstellar medium. I won't go into all the details, but just to tell you, in different regions of the interstellar medium, you have different temperature, different densities, and different state of hydrogen. Different state of hydrogen, what do I mean? Hydrogen can be existing as a molecule, H2. It can be H plus or H. So different regions have different weightages of this different structure of hydrogen. And what we astronomers try to do when we try to detect molecules, we try to associate the detection of molecules with any of those regions. What I mean is, do we find, let's say, carbon monoxide only here or everywhere? So we try to do that, And by doing that, we try to understand some aspects of the evolution of the molecules. So what I'm going to show you now, because it's easy to say molecules have been detected, but we are really sure in the scientific community that that those molecules have been detected unless you have a research publication in some high impact factor journal talking about it. So here, what I've done here, I've taken some journals during the last 4 or 5 years, which have reported the detection of the molecules. So there's one in 2014 in Astrophysical Journal, where they show you the molecules, 39 molecules have been detected by the Herschel Space Observatory, which is found in a satellite outside, beyond our atmosphere. And they have detected ionized hydrogen plus 39 molecules. So those pictures, as I said, they are just to confirm what I said before. So this is a picture of Crab Nebula, which is basically a star which has exploded like a supernova, and what is left is called a supernova remnant. And for quite some time we thought, for different scientific reasons, that molecules cannot be found in supernova remnants. So that was quite a pioneering paper which showed that molecules can be detected in supernova remnants. So it's called the Eskimo Nebula because, you know, we astronomers, we give names according to what we visualize. Because it looks like the head of an Eskimo, we call it Eskimo Nebula. And some very complex organic molecules have been detected. A paper published last year. It's a Horsehead Nebula. You can see the head of the horse. So it's a region where a lot of stars are being formed. And I'm just showing the molecules which have been detected. So it has been detected by a radio telescope because the spectral lines happen to be in radio. So Eagle Nebula. So you can see those kind of cloudy stuff here. So at the surface of those clouds, you have stars which are born. And you do have some American university, which I've forgotten, which have taken pictures of those nebula every 2 years during the last 15 or 20 years. And they have been able to make a movie to show how the star moves from the surface to outside. So in all of them I've given references. So it's basically some region in the interstellar medium. This has been published in monthly notices of Royal Astronomy, where they have seen some very complex organic molecules. So this is part of the slides which I will skip. So there's a branch of chemistry. We can call it quantum chemistry, gas phase chemistry, or solid state chemistry. So I will skip the slides, but I'll just tell you briefly what they try to do, computational chemistry also. So they try to come up with models, very often computational models, to explain the formation of the molecules. And they've done a huge amount of job, but unfortunately we are still limited with amount of data concerning the molecules. So different mechanisms to heat the interstellar medium, which I won't elaborate. Different cooling mechanisms in the interstellar medium. So just to tell you that it's very important to know all the mechanisms which can heat or cool the interstellar medium. Otherwise you will never be able to understand how the molecules form. And you should understand the kind of reactions which you have. I've written some here. Yeah. So maybe, how many of you have done physics at O level, A level? Most of you? Okay. Maybe you know that if you take an atom if you use quantum theory or quantum mechanics, you know the energy levels are quantized. And when an electron comes from a high energy level to a lower energy level, like let's say EM minus EN, you have a photon of a very specific wavelength which is emitted. Now, to understand those molecules, the energy levels which I've been talking about here are not so important. What is important, it is, again using quantum mechanics, we try to understand the rotation of the molecules. So you have different types of rotation which gives different energy levels. So then we have what we call rotational spectra. So what the telescopes observe, it is mostly the rotational spectra, but also vibrational spectra. So each molecule has some modes of rotation. Each molecule has some specific modes of vibration. And each mode of rotation and vibration, they have different energy levels. So when you have a transition in energy, a given wavelength or given spectrum is emitted. So this is what we observe in astrochemistry. So it is observed by different telescopes, can be radio telescopes, millimeter telescopes, or infrared telescopes mostly. Yeah, now just one aspect about the lines we observe. How many of you have been caught by overspeeding on the road by cameras of policemen? None of you? Oh, you're good drivers. So the camera uses the principle of Doppler effect. So basically, if you have a relative motion between the observer and the person detecting the signal, then you have a change in wavelengths. From that change in wavelengths, you can find the velocity at which you are moving. It's what the policemen do to stop you on the road if you are overspeeding. So what happens when we are looking at those different regions in the interstellar medium and different galaxies? So nothing is static in the universe, everything is dynamic. So because there is some kind of motion, and because of that velocity, the spectral line we observe will be shifted. So we call it Doppler effect. So before observing a line, it is very important to, by independent methods, to estimate that velocity, to find out in that given region, what line should I observe. So for different regions where different velocities, where you have different velocities, you have to do that calculation before observing the lines. So I'm not going to explain you what you have on the slide, but just to show you that it's not an easy job. I would even say it's a complicated job in designing the hardware, the software to detect a given line. Just to show you that my job is not so easy. Now some pictures of the telescopes to know that what I'm saying is real. So this is the biggest steerable dish in the world. It's called Green Bank Telescope, which has a diameter of 110 meters. So it's found in a place called Green Bank in US. So basically, the molecules which you want to detect in space, the radiation they are emitting is very weak. So to be able to detect this radiation, you need a huge collecting area. So if you have a dish, let's say, of 10, 15 meters, you will not be able to detect molecules. So you need huge dishes. So this is one of the most important radio telescope, which is a single dish which can move to detect molecules in space. So it's just some research papers to show you some molecules which have been detected by the Green Bank telescope. So So anyone interested in those publications can contact me after. Now you can combine different dishes to produce much bigger collecting area, and which can make a telescope which is more powerful. So this is one of the most powerful radio telescopes in the world, which is found in the state of New Mexico in the US. You'll be happy to know that one of the pioneers of astronomy in Mauritius, like me, his name is Dr. Kumar Gulab. So he's got American nationality now. Unfortunately, there are no challenging problems for him to come back to Mauritius, so he's working in the VLA. So he has used the VLA to detect some molecules. Again, some research papers which tells you some molecules which have been detected at the VLA. Now you have a new type of telescope. As I said, it measures wavelengths between infrared and radio. So they correspond to frequencies above 30 gigahertz. They can go up to 200 gigahertz, which is found in Chile. It's one of the most powerful telescopes in the world. It's called ALMA, which has detected many molecules. So one of the most recent detections was glycoaldehyde, which is a molecule known by Sabrina. Have you heard of? So I'm glad to tell you that it has been detected recently, and it has been redetected in 2013 and 2014. Now, if you take, I think Dr. Gopi was asked me, each molecule, so each molecule has hundreds or thousands of lines. So one of the problems of detection, you can have one line from a molecule and another line from another molecule which are very close. So if the telescope does not have sufficient frequency resolution, those two lines will merge. Then it's difficult to say from which molecule it is coming. Then you need to have other techniques. So the SK project, which Kamlesh has been talking about, will have a frequency resolution around 100 times better, which means because of thanks to the SK radio telescope, we'll be able to distinguish between the lines which we would not have been able to distinguish otherwise. So it's just an example of, so each line here corresponds to a given molecule, which is a spectrum of one of the spectra of a given molecule. This is the most powerful radio telescope in the southern hemisphere, which is found in Australia, which has been widely used to detect molecules. Again, some molecules and some publications which have been made using this telescope. There's a website which is called the SPLAT-TALO database. Those who are interested to know about all the molecules which have been detected so far, and by which telescope and which spectral line, you go to the database, you will know. So if you're not sure whether a molecule has been detected, you go to the database. If you want to know whether a molecule, then it will help you to try to see if you can find a new molecule in a given region. So anyone studying astrochemistry must use this database very often, otherwise we will be reinventing the wheel. Now, molecules have not been detected only in the Milky Way. They have also been detected in neighboring galaxies, like the small and large Magellanic Clouds, the Andromeda Galaxy. So with the SK, we'll be able to go further, detect molecules in other galaxies also. So when you detect prebiotic molecules in other galaxies, it can give you a lot of ideas about the origin of life. So it's a typical spectra of molecules. So we call in astronomers, we call it a forest of lines. So basically, depending on the frequency resolution you're using, you'll get a lot of lines. Then you'll have to first find out where it has been observed, do that Doppler correction, and then from computational chemists, find out which frequency it is and to which molecule it corresponds. So whenever you have such a forest of lines, in most cases, you have some lines which are not identified. I think it's written here. Like this has not been identified so far. This one also. And one of the research which astrochemists try to do is to try to find the origin of those lines which have not been identified. This is another example. So you don't observe the spectrum of one molecule. So the spectral line receiver, depending on its frequency resolution, will give you a forest of lines, and you have to dig in and try to conclude which lines have been observed. Sorry, which molecules have been detected. So briefly about the SK. So the SK is going to be the most powerful telescope in the world, which has started to be built. So the countries involved will be South Africa, which will be like the father figure, which the project has already started in South Africa. It's in a place called the Karoo Desert, where 4 dishes have already been set up. Then you have Namibia, Botswana, Mozambique, Mauritius, Madagascar, Zambia, Kenya, and Ghana. So you'll have the picture you can have in your mind, you'll have thousands of dishes. Now, whether it's going to be 3,000, 4,000, 5,000, that has not been decided yet, but it's a few thousands of dishes. And all those dishes, most probably it's also not been finalized whether you have 4, 5, or 10 dishes in Mauritius, but definitely you'll have 5 to 10 dishes in Mauritius. So all those dishes will be connected, and all of them will be looking at the same region in the sky at one go. So that will be the most powerful telescope in the world. It will be more than 50 times more sensitive than any existing telescope. And the technology to be used in this telescope still does not exist. So you'll say, am I crazy? What am I talking about? If you look at the progress of hardware and ICs, okay, they follow a certain curve. And so far, it has not failed. So we believe that in 5 or 10 years' time, we'll get the computing power which we need to analyze this data. So the data which you're going to collect, it's going to be huge. And our human mind only will not be sufficient. So nowadays, there's a field which is bringing a huge revolution in technology. It is big data. So now, big data, the advantage of big data, whatever algorithms you develop in any branch can be used in other branches. I'll take an example. The software we developed to convert the binary data from a radio telescope into radio images, the same software is used in medical imaging. So that's why I'm saying big data will be like the link with different branches in science. So definitely, whatever I've been talking about in all those issues and puzzles, a lot of those issues and puzzles will definitely be solved by the SK. But that will take time, next 5 years, 10 years, or 20 years. So my conclusion is that observations of complex organic molecules, so we need not wait for the SK. There's still a lot of work to be done using ALMA, GBT, and the other telescopes to observe in more distant galaxies, and a lot of research work in chemistry, physics, astrophysics, even computing, big data have to be used to understand more about astrochemistry. So thank you for your patience.